service teacher attached to the Anogena Center and the Science Major Teacher Training. My name is Miss Ovita Persaud. So good afternoon, students. How are you this afternoon? You seem as happy as I am and ready to work. So before I begin today's topic, let's recap last class work. So last class we discussed proteins, testing for proteins. Now can somebody tell me what they have learned about proteins? Yes, Mary. Proteins are an essential diet? Correct. Okay, yes, Patsy, let me hear you. Right, that's correct. Proteins are made from a chemical called amino acid, and it's an important part of a healthy diet. So, class, today we'll be looking at another source of nutrients. That is, testing for starch. Now, does anybody know what is starch? Well, you cannot tell me no because we all know that we use starch in our everyday life. We plant starch, we eat starch. Some examples of starch are John, rice, very good. Mary, sugar, no, that's wrong. You can try again. Flour, yes, that's correct. Yes, starch. Starch is the main source of carbohydrates and it's an essential part of our healthy diet. Like I explained, starchy foods are main sources of carbohydrates. On the board, there's a definition for you to read. Read with me. Starchy foods are our main sources of carbohydrates. Now, so on the board, I have for you a little activity. I want to know if you know what are the lab equipment we are about to use for our experiment. So I'll call on you. You're going to stand and tell me if it's correct. Here we have 3 labelings. I will call on 3 students. You are to stand and tell me the correct labeling for 1, 2, and 3. Number 1, Jason. Please tell me what is the name of this showing on the diagram. Pipette? Correct. Give them a clap. Number 2, Peter. Petri dish. That's correct. Give him a clap. Number 3, Robert. Food samples. That's smart. Give him a clap. Good job. You all understand. Give yourself a little clap. So we'll move on. So any question? No? Okay, I have a little activity for you. We have some starch samples and we're going to be testing for the presence of starch. Yes, correct, starch. We have 3 sets of Petri dish, label A, B, C. There are food samples in each. As you can see, chana, pasta, flour, rice, corn, and here we have milk. This is our materials, iodine solution, and pipette. So we're going to be carrying out this experiment. Now we are going to add 2 to 3 drops of iodine solution to each food sample for 1 to 2 minutes, and then we're going to record our observations. After 1 to 2 minutes, we are back to see our results. Here we have our chana, it turned blue-black. Our pasta turned blue-black. Our flour turned blue-black. Our corn turned blue-black. Our rice turned blue-black. But our milk remained orange. So, from our experiment carried out, starch is present in all the other food samples except milk. Yes, that's correct, John. So I have an exercise in hand for you which stated, from the experiment conducted, state if starch is present in the food samples below. We have rice, corn, chana, milk, pasta, and flour. You are to state whether starch is present or not. After this exercise, students, before you begin this exercise, I just want to let you know that after this exercise, we will be looking at a video based on the importance of starch in our bodies. Okay, students, I'm very happy that all of you understand. On the board, I have the correct results from the experiment conducted. Please copy in your notebook. Also, is there any question? No? Well, I have a question for you. How do you know when iodine solution is added to the food samples? How do you know if starch is present? Jason? Yes, that's correct. When it turns blue-black. So I have homework. I want you to read up on testing for facts. Enjoy your afternoon.
Of carbohydrates. The absorption of simple sugars occurs in the cells of small intestine. The cells of small intestine are called enterocytes, which can only absorb sugars in monomeric forms such as galactose, glucose, and fructose. The apical side of enterocytes points towards the lumen of the small intestine and contains digestive enzymes to break down disaccharides into monomeric forms. Below the basal lateral side of enterocytes, there is an interstitial fluid which separates the blood vessel from the enterocytes. The sugars undergo a different transport mechanism. Glucose and galactose enter enterocytes through sodium glucose transport protein 1, sGLUT1, and fructose enters enterocytes by sodium-independent GLUT5, integral proteins that facilitate passive diffusion. The basal side of enterocytes contains a transport protein, sodium-potassium ATPase, which uses ATP for the transport of 3 sodium ions outside the cell and 2 potassium ions inside the cell. An electrochemical gradient with a low concentration of sodium leads to the transport of sodium ions from the lumen into the cell through a hexose cotransporter protein, sGLUT1. Along with the sodium ions, 6-membered hexose sugar molecules like glucose and galactose enters the cell through the cotransporter. The fructose does not pass through sGLUT1 due to 5-membered sugar, and it uses an integral protein transporter, GLUT5, and enters the cell through passive transport without the aid of ATP. Inside the cell, the fructose molecules are transformed into glucose. The molecules like galactose, glucose, and fructose leave the cell through a passive transporter, glucose transport 2, GLUT2, and enter the bloodstream. The glucose in the bloodstream eventually enters the liver and stored in the form of glycogen in the liver cells.